All right, we're going to go ahead and get started with the rest of the program. So welcome back, everyone. My name is Jillian Shaw, and I'm the Associate Director of the Ladders to Cures Accelerator. It is my absolute pleasure to welcome a fellow Brody, Juliana Gentile, to give a talk today representing Sonia Valab and Eric Minical's lab and her work on prion disease. Juliana actually did her PhD at Yale, focused on biomolecular physics and biochemistry, and studied actin remodeling in dendritic spines with a lot of implications for CNS disorders. Today, Juliana works on in vivo biomarker discovery and has a really elegant Divalent siRNA for prion disease story to tell you all about. Welcome, Juliana.
So like was introduced, today I'm going to tell you about our Investigator-Initiated program for Divalent siRNA for prion disease. So this is Sonia Valab and Eric Minical, who co-run our lab here at the Broad. They started on this journey back in 2011 after Sonia's mother passed away from a genetic form of prion disease, and upon further genetic testing, Sonia learned that she too had inherited the mutation. This led to Sonia and Eric leaving behind their current careers, retraining as scientists at Harvard Medical School, and starting our lab here at the Broad in 2019. Over the years, our lab has grown in numbers, and currently we're 16 research scientists, associates, and computational biologists, but the core founding principle of finding a cure for prion disease has remained at the forefront of our mission. So prion disease is a group of rare neurodegenerative diseases where there are about 500 new cases in the US every year. Most people sitting in this audience have probably heard of mad cow disease, which is an acquired form of prion disease, but today, acquired cases account for very few new cases of prion disease. More commonly, prion disease is sporadic or genetic, and this disease is universally fatal, where most patients live only 5 months from symptom onset. And despite all these differences in names, etiology, and early symptoms, the genetic blueprint of this disorder is really simple at the molecular level. So every single one of us expresses prion protein, also called PRP, and under normal circumstances, this protein is harmless, but different factors, be it external, genetic, or spontaneous, can cause this protein to misfold. This leads to a cascade of misfolding events to the normal PRP around it, leading to cell death and ultimately rapid death of the patient. So our therapeutic hypothesis revolves around the idea that by getting rid of the good protein, it will never have the chance to misfold. And so today I'm going to focus on our approaches that target the RNA for degradation, ensuring that the protein isn't made in the first place. And maybe you're sitting there asking yourself if this is a safe thing to do. And the answer is yes. There's been many proof of concept studies showing that PRP is dispensable for healthy life. If you knock it out in different animals, these animals are completely healthy. And the thing that's more reassuring in PRP knockout animals, if they're challenged with prions, they're completely resistant to prion infection. And therapeutically, we've also shown that this is a safe and effective strategy. So in this survival curve, when you challenge wild-type mice with a strain of prions, they routinely are seen to die around 160 days after infection. But if you lower prion protein by 50% by dosing an antisense oligonucleotide early in the disease course, then you see a large survival benefit. But what you can also see from the survival curve is that all animals eventually develop symptoms and die from the disease. So ASOs were the first treatment for prion disease that we were able to help bring to the clinic through a long-standing partnership with Ionis Pharmaceutical, but we'd like to have as many shots on goal for this disorder. So this means that looking forward, we had a few benchmarking goals for the next generation of PRP-lowering drugs that we bring to the clinic. With ASOs, we were only ever able to drive PRP knockdown to 50%, so we wanted to find modalities that could drive knockdown even deeper than this. We wanted molecules that were durable and required less frequent dosing if needed to be dosed in a chronic paradigm. And we also wanted to find modalities that could distribute uniformly across the entire brain, since this is a whole brain disorder. And finally, we wanted things that were safe and well tolerated. So we kicked off an exciting new collaboration with Anastasia Kurova's lab at UMass Chan when they demonstrated that they could modify siRNAs in a very beneficial way for brain delivery. So when a traditional siRNA is delivered into the brain, it has very low target engagement. And here in the Cy3 labeled image, you can see that a traditional siRNA also has low target engagement. So what Anastasia's lab did was she linked two siRNA molecules together with a flexible linker, joining the passenger strands of these molecules together, and showed much broader CNS distribution. These molecules were potent in knocking down their target RNA, and we were really excited to develop these as a new therapy for prion disease. So first, we screened just a limited number of mouse targeting sequences, and we found a tool compound that lowered PRP by about 50% in the whole hemisphere of a mouse brain. We can also ask how these molecules work in the disease model, and so we dosed these into wild-type mice challenged with RML prions in an early symptomatic time point when mice have just started demonstrating symptoms and have just started to lose weight. And we see that when you give a single dose of this divalent siRNA to these animals, they survive 3.5 months longer than the untreated controls. We also took this tool compound into a chronic dosing paradigm and dosed it every 120 days, starting at a pathological time point, meaning that neuroinflammatory markers in these animals have risen, but they don't have any overt symptoms. And we see a 2.7-fold increase in survival, and this also delayed symptom onset, and symptoms were also slowed in these animals. So these survival curves probably look pretty similar to the first ASO one that I showed, but like I said, this was a very limited screen that we did to find these mouse targeting tool compound. And so we knew that with deeper screening, then we could find a molecule that led to deeper knockdown in these animals. And so what I did was I screened 84 compounds in a human cell line, as I'm showing here in this plot. And at a 0.5 micromolar dose, we were able to discriminate the most potent sequences. And once we confirmed that these were active in dose response, I was able to take the most potent 7 sequences forward in vivo. And so we tested these in a mouse model that has the mouse gene knocked out and re-expresses the human PRNP gene. And what we could see across all sequences that we tested and scaffold combinations in the whole hemisphere, 2439 was superior. And I'm not showing the RNA knockdown data here, but I also dissected out individual regions of the brain and showed that in all regions that we looked at, this was a superior sequence. We next asked how durable these oligos were, and so this I am dosing half as much of the 2439 as what I showed on the previous slide in our humanized mouse model. And I took animals down at 1, 3, and 6 months with undosed matched controls at these time points. And what we see is that at 3 months after just a single dose, we retain 50% knockdown of PRP in the whole hemisphere, and some activity is still retained out to 6 months. So how does this stand up to ASOs? ASOs need to be dosed higher, and their durability is basically gone by 3 or 4 months after a single dose. And so we were pretty excited that these molecules were more durable than things we had tested previously. And so kind of taking a pause at this point, what I've shown you up until this point, screening, proof of concept in vivo and in a disease model, as well as durability, is what is typical for an academic lab to lead. And usually at that point, it transitions to a company to lead manufacturing, GLP toxicology, and filing to the FDA. But we were in the unusual and fortunate position to bring all these things forward ourselves. So first, we produced a small GMP batch of 2439S4, our lead sequence and scaffold for toxicology as well as clinical studies. And given the severity of our indication, early on we went to the FDA and asked them if we could do only rodent toxicology studies, and they said that we needed a non-rodent model. So here I'm showing our dog toxicology data. So these animals were given a single intrathecal dose administration of 20, 60, or 200 milligrams, and were necropsied at 28 days after this injection. And upon necropsy, we found no adverse findings, both in the dogs and rats that we did toxicology in. On the biodistribution plot, I show an estimated IC50 of 1.2 micrograms per gram, and this is estimated from PD-PK data that I have in mice. And we see that drug accumulation close to the site of injection down the spinal cord and in superficials of the brain. We have more drug than our estimated IC50, meaning that we would probably have meaningful knockdown in these areas of the brain. But in the deepest regions of the brain that fall below the IC50, we probably still have poor distribution of these drugs, similar to what we saw previously with ASOs. So this kind of wraps up all the data that I was going to show you today, and kind of I wanted to summarize at this point where we're moving now. So here pictured is the actual drug vials that are ready to be injected into patients. We have had our IND cleared by the FDA, and we're going forward with leading a trial supported by NIH. And so first, our plan is to dose 15 symptomatic patients with a single dose and just assess safety and PRP lowering in CSF of these patients. We're currently working on getting all the trial sites up and going, so we have 5 trial sites across the US for this. And we are also working to get a pre-symptomatic arm for a trial up and running later in 2026, and this would also be a single dose. And so moving forward to a multi-dose study, we first need to perform the multi-dose toxicology studies. So just zooming out, this whole journey started not too long ago, and I think all of us are really proud of where it's come in such a short amount of time. We are really excited to bring a first-in-human to the clinic for our disease. And we're also excited to be able to share this knowledge broadly with patient population as well as the broader field, also working on siRNAs. So with that, I would like to thank all the people in our lab that helped this project come to the point it's at right now, our collaborators at UMass Chan that helped with, as well as at Atlanta Therapeutics that helped with synthesis of oligos for the preclinical studies, as well as all the CROs that have helped us throughout this whole process to manufacture as well as run the toxicology studies and our funding along the way.